Today you will hear again from Dr. Mukesh Jaiswal, who is one of the application scientists, works in the areas of next generation sequencing technologies. He will talk to you about whole exome sequencing kit for investigating rare diseases. This is probably going to be the last lecture on the NGS technology and its application. And while today's lecture is not going to cover much of the basics of NGS, but is definitely going to give you more information about possible applications from these platforms. So, let us continue on this lecture today, and then we will try to conclude what we have learnt out of this NGS based platform from basics to the applications.
Okay. So, today we are going to talk about investigating the rare disease and its treatment with the Agilent solutions. So, so I am going to cover about what are the rare disease and how basically it can be diagnosed by the NGS solution, and how we can basically give the treatment to the patient, right. So, we have some solutions where basically you use Agilent solution for the diagnosis of rare disease and its treatment part. So, it's going to cover some background of rare disease, and then I'm going to talk about some part of CRISPR-Cas, how basically it can utilize for the treatment purpose. So, what are the rare disease? Rare disease basically is a rare disease. It's affected 60 million people in US and Europe. So, it's a big number, it's quite a big number. And 7,000 rare disease basically known till now, and 80% reason of that is genetics. Is something wrong in their genetics. So, yes please.
We are talking about 60 million affected in the US and Europe.
Yeah.
Is there any study what anybody
And yeah, I'm coming to the next slide. So, I'm coming to the next slide. So, this is like some worldwide I'm telling. So, now I'm going to the next slide. I have Indian data also. And 50% affected children are basically affected with this disease. So coming to Indian scenario, yeah. So, ICMR, the Indian Medical Council of Research, launched this registry in 2017. And they said that around 70 million people in India is also suffering for this rare disease. So, it's a quite big number, and that's why they launch a project, ICMR registry, where you can basically write a grant to them to work on the rare disease, what's the problem, how you can diagnose that thing, what's the treatment part of that. So, in April 2017, they launched this project, and so these are the key objectives of this ICMR registry. So, its main objective is that to understand what's the problem of rare disease, what's the causation of that, and how this data can be utilized for the treatment of the rare disease. So, these are two main objectives of the ICMR, and so it launched, and I think it's available for the grant application also. Okay, so I'm going to talk about some of the rare disease which is existing worldwide. Is worldwide. So let's come in the first, cystic fibrosis. This one. So this is a disease basically where the excessive mucus is deposited on lung or pancreas, which cause the respiratory failure and inability in the digestion part. And the median survival rate for this disease is 40 years, right? And the worldwide is 70,000 patients are known worldwide. Then leukodystrophy, this one, and this is again the progressive disease affect the brain, spinal cord, and the nervous system. And this is basically children basically affected with this disease, and 5 to 10 year basically children, and around 60,000 children are affected worldwide. Retinopigmentosa, this is another disease, and it is affected. It causes the blindness and it basically the survival is like 40 year, and around 100,000 or 15 million, 1.5 million worldwide patients are known for this disease. So what's the cause of this? What's the cause of this? The cause of this basically, sometime it is affected by the single gene, or sometime it's affected by the multiple gene, right? So if it is like the first one, cystic fibrosis, is only the one gene, that is CFTR. There are several mutations on CFTR, and then this transmembrane transporter is basically disturbed, and that caused the cystic fibrosis. So in this cystic fibrosis, only one gene got affected, right? But if you see these two, the multiple genes are affected. So it's very difficult to identify when the multiple genes are affected. So here, like 30 genes are affected here, and in this disease is 77, right? So problem is that this caused the damaged neuron, and here in the retinitis pigmentosa, it is cone cell and the root cells are basically disturbed, right? So these are the disease, and there are multiple genes are basically involved in there. Some more, which is included in the ICMR project basically. So this is childhood ovarian cancer, ovarian cell carcinoma, endometriosis, and there are multiple genes basically involved in this disease also. So this is also incorporated in a ICMR registry. You can go in their website, you can basically look what are the rare diseases. The challenge is that is their diagnostic, how to diagnose this disease, right? So if you go the regular process of diagnosis of this one is pretty expensive, and doctor basically takes at least 8 years to diagnose, and they go like 40 different methods to diagnose this test. So because the complexity of disease is not like one gene, it's like multiple genes they are affected, right? And basically they take at least 7 years and 40 methods they use for the diagnosis. So that's a challenge. It is pretty expensive, it takes time. But for the treatment purpose, if you have early intervention of this disease, you know the cause of very early, then the very early diagnosis, then you can do early intervention and then improve the quality of the life. So this is a challenge, but if it take a lot of like 8 years to diagnose only, it would be difficult, right? So that's why it is very important to diagnose the disease in very early stage. So right now, if you see this rare disease, now 2012 when the exome panel just started, it was only 130 gene. Now 2017 is more than 200K mutations are known for the rare disease. Basically, this is because of the more advancement of the exome panels. So you need to extract the DNA from the patient, right? And then go for library preparation, and then target enrichment. After seeing the library is basically perfectly fine, you can sequence and go for data analysis. So this is kind of one workflow. Basically, you can use our exome panel for the diagnosis of the rare disease. So the challenges is always there, but I would talk about why the Agilent's exome panel is more better actually in sense, because we make RNA baits, and it is oligo baits basically, and these are because we make the RNA bait, they have the better RNA DNA hybridization, and these are the high fidelity bases which we make by the inkjet technology. We make this bait by the oligo. So this is our high fidelity bases. If you see the error rate in the base in the probes basically is very low in Agilent. It's like 1 or 2 error basically in 1 kb. But others have lots of so we have the high fidelity probes basically which are biodynamic, it is used for making the libraries. So this is the different ways you can make the exome libraries. So starting material always with the genomic DNA, right? We have 3 different ways to make the libraries for the exome sequencing. One is XT, XT2, and QXT. XT is basically is this one. If you have different patients, 8 patients, right? You can make an individual library from each patient's independent capture, and you can pool while sequencing, right? When you go for sequencing, you can pool this sample and go for one sequencing run. So that's the XT preparation of the exome preparation. Another, if you want to do some comparative study, you can barcode the patient sample and pool itself, 8 to 16. In one pool, you can follow by the capture and go for sequencing. So you can compare between the patients also, that is XT2. And another is library preparation is based on enzymatic sharing. So you can use transposase enzyme to make the libraries basically. Transposase enzyme, then hybridization followed by the sequencing. This is the fastest way you can make the libraries for the exome sequencing. Okay, so performance for all these methods to make the exome library is pretty good, and they get very good coverage, more than 95% coverage all the methods. This is the 3 pillars basically Agilent works on, basically performance of the exome library, contents, and the flexibility. We work from decades to improve the performance, content, and the flexibility of the kits basically. So if we see most of the rare disease basically now studied by this panel, is called a clinical research exome panel, and this exome panel contains all the exon region and also the panels on the intronic part of the region, which basically associated with the inherited disorder. We have the latest exome panel V7. This is mentioned V6. But we have now V7, and that basically covers all the translational and the clinical research panel. It covers whole exome. Another, we have the very small panel, the focus exome, which is covered the disease-associated region. But most of the rare disease, basically, which I talked before, it is basically used for the clinical research panel. So if you see the contents performance of this one, with this clinical research exome panel, they have like 5,000 gene is basically deeply covered with the disease-associated region with the clinical exome. And the challenge is that when you go for the diagnosis of rare disease, the most of the mutations are present in the GC region. To make the library for the GC region is always challenge. So, but with our clinical exome panel, the performance in the GC region is very good. It's very uniform preparation of the library when you go for the GC preparation. So content basically, what's the content for the probes basically? It is basically designed by the Dr. Madhuri Hegde from Emory University, and they make basically the probes which is covered all the disease-associated region, exon, and intronic part, which covers maximum rare disease parts. For example, this is the leukodystrophy, right? And this pathogenic variant, that is JC2, this gene, basically if you see, compare, because this is the GC-rich region, and if you compare with the other vendor, you don't see any coverage. There's no coverage for this gene. And if you see the clinical exome panel, we covered this part also to be able to detect the 5' UTR variants with this disease. So in this disease, this is the kind of pathogenic variant, and you can easily detect by the clinical exome panel. Another, retinitis pigmentosa. If you see 2 other vendors, this CFD1 gene is not covered in this part because this is intronic part. These are exonic part. Intronic part are not covered. right? But if you see our one is fairly covered, that part, that means is detection of that mutation is very easy on that. Then if you see this region, again, is well covered by this, these are all non-coding region. These are well covered with our clinical research exome. And most of the pathogenic variant basically for rare disease is present on non-coding region. So it is fairly covered with the clinical research exome panel. So if you see overall in the clinical research panel, we cover all the clin variants, pathogenic regions, and it covers mostly like 98% regions are covered. And others basically has low coverage. So if you think about when the doctor is going to diagnose this leukodystrophy, it takes 8 years, right, by the normal method. And the average test basically they do around 20-30 tests to diagnose for this one. It takes 8 years, right? And the cost goes up like it's like $20,000, right? But when you do one simple test, our clinical exam panel you easily identify this mutation, and that is basically for the leukodystrophy, and it is very cost effective, right? And
Cost of one, let's say there's a patient who would like to get that.
so it's just going to cost like 15,000 rupees, right? So one test costs like 15,000 rupees for exam panel.
There's a chance that there might be some rare, there is just a chance that there would be some rare disease that is detected.
Yeah, because it covers the intronic part, exome part, definitely it's going to detect that thing. But detection rate is faster, right? And it takes less time. So doctors start their intervention much earlier. With that, like suppose this is our content, but we doesn't make this content in vacuum. We do the research and we make the probes for cover all those reasons, right? But sometime when you did your experiment, right, and some part basically you think that this is the part, maybe pathogenic part, and it is missing, right? And you want to incorporate that part in your panel, so it is very flexible. We can customize the panel according to your requirement also. Suppose any gene and it is not basically the intronic part is not covered, and if you want I want to cover this part also, we can basically add this panel and incorporate in your panel. So that's our flexibility. So we work on 3 parts: performance, and the contents always optimize year by year, and then flexibility if you want to add more, right? So it's very simple workflow that I told. Start from the library preparation, then we make the targeted panel by the probes, right? And then data analysis and reporting. Whole workflow basically takes 3 to 4 days, and it's easy to identify the kind of challenges for a diagnosis of rare disease, right? So now this part is kind of over. Like if you get some kind of mutations, right, in any disease, not only in rare disease, suppose cancer, right, and but it is multiple mutation, and you want to solve this problem for the treatment purpose. So we have a tools called as the CRISPR-Cas, where basically you can do the gene editing, right, and to fix that gene for the treatment purpose. But this is very early. We launched some libraries for the treatment, but it's very early stage. Let's start with that. What is a CRISPR-Cas? This allow you basically to allow the mutation to correct maybe, or gene editing. So it is based on the guide RNA, right? So this is the guide RNA, this one, and this is the site of recognition, and this is the Cas9 enzyme. When this become active, they attach, and it goes to the PAM site like this. If it is identical or hybridized on that, like this, it is identical with that, it creates a nick. right? This Cas9 enzyme, when this guide RNA is specific to that, it creates a nick. Now it allows you three possibilities. One possibility is that just leave like this, and body repair, body system, our cell system basically go for non-homologous joining and cause the knockout of that gene, right? It creates the knockout. Second is the homologous recombination. If you want to do the gene editing and you have homologous sequence, you can incorporate this homologous sequence in that, right? Third part, this is the deactivated Cas9 enzyme. This yellow color part, it might be activator or deactivator. Depending upon the activator, it can induce the gene expression or reduce the gene expression. So it allows the whatever mutations basically you got from the exome panel, right? You can basically try to correct, or you can do the gene editing for their treatment purpose. So that's whole story, exome panel, and what's their follow-up. So we are working on the functional genomics, where basically we can try to edit these genes in a large scale, like, and it might be any knockdown, knockout, and knock-in, anything, and try to edit their genes to solve the problems. So this is the, if you study the functional genomics, so very first prospect is knockout, right? If it is knockout, means guide RNA breaks that one, it makes the truncated protein, right? So your protein not going to work, it's truncated protein. Knock-in means is basically going to add some tag on that, on the protein. Turn off means if it is higher expression, this is the repressor fusion. If it is turn off means the lower expression of the gene. This is the genomic mutagenesis. Here basically it's a site-specific mutation you can create by the CRISPR-Cas. Suppose you got some mutation and you want to solve that mutation, right? You can change a base by base by the mutagenesis, right? So this way you can correct that SNPs you got, right? You can correct that part. So this allows you to site-specific changes with the Cas9. And if you want to do some genes are basically lowly expressed in some disease, you can basically induce the expression to the higher level, right? So it can induce the gene expression. So you can do multiple function by the Cas9. You can induce the gene, you can repress the gene expression, or you can do the site-specific changes in the gene, right? That's allow you 5 different possibilities to do. So now the question is that, suppose you like in cystic fibrosis, right? You work on single gene, and there may be 1 or 2 mutations you got with the exome panel, right? And you want to fix that problem, right? So there's 2 ways. One way means if one gene and few mutations, so basically you're going to use 4 to 5 different guide RNAs, not more than that, right? So for that one, you can make the guide RNAs in your lab. So suppose this is the target basically. You're going to make a guide RNA for that one only. So this is the target panel, target you want. Just add the T7 promoter on that, right? If you add the T7 promoter on that and go for, so the kits basically what they have the T7 primers and go for in vitro transcription to make a guide RNA. For in vitro transcription, you make a guide RNA which is the reverse complementary to the target, which is going to be reverse complementary to the target. So this is the kind of guide RNA. This is the red part basically is very specific to the target to the part where you're interested for, and this is the backbones, minimum backbone. And this, when this going to bind on the target gene, this targeted part, in the presence of Cas9 enzyme, this gene basically cleaved in 2 parts. If you verify this one, in the presence of Cas9, this gene basically cleave off, right? So this allows 5 different possibility again. Basically, going to maximum time is going to knock out the gene, right? So if it is your genes in numbers as 1 or 2 gene, and you want to use the, you can make a guide in your lab also, right? But mostly, it's not a case. It's not a case. There are multiple genes are is involved for any complex genetic, right? Like retinal pigmentosa, 77 gene is involved, and mutations is like more than 1,000 mutations are basically involved there, right? So what we do, we make a guide RNA for that panel, right? And this is totally custom thing. Is we don't make, we used to make some catalog, but we used to totally depend on the users. So we make a guide RNA on a slide. We cleave that one from the slide, do the PCR amplification, and packed on the viral particle. Now it's ready to transfect in the cell system. That much guide RNA basically, which is for that mutation, right? It's going to ready for transfection, and you can use for the therapeutic opportunity for this. Right? So this is
That, so the target cells are actually cell culture.
yeah, this is cell culture.
And then
So it's a viral particle. These just cells basically to transfer directly into there.
cells are then
Transfect with this guide RNA. They already have a Cas9 enzyme, so it creates the, means Cas9 is there, right? So it's going to cleave that part of the gene. Or it's depending on what you design basically.
so those cells would be treated.
Yeah.
Then how does it go? How is it taken forward from there?
So see, I told you this is very early stage. Again, to do this thing, you need to do lots of screening, lots of NGS work to identify it's going to work or not. So this is just a thought, and guide RNA is we can synthesize for you, but again, the protocols and how it's going to translate to the actually patient is in very early stage. If it's like 10-20 gene you can make in your lab, but if it is more than 1,000 gene in high scale, then we can make. Means it's for the high scale, not otherwise you can just make in a lab. Just your any target at T7.
Specific features of it. We know that these are specific targets.
Yes, specific target. Just add the T7 promoter on that. Use T7 primer, and do in vitro transcription. That's inside the guide RNA. So it's very straightforward protocol. But yeah, again, you cannot make the 1,000 guide RNA for multiple gene, right? In that approach, you can use these guide RNA strategy and try to solve the problems. But yeah, I would tell you this is not easy. It's very, very hard, right? It's very hard because if you see this is going to be like thousands of the guide RNA and going to transfer in the cells, right? Now you're going to go give some treatment by drugs, right? And then you need to go for screening protocols day by day, and again you're going to verify these things going to be work by the validate or not by the NGS. But of course, this is the thought, and you can basically try to solve that problems. So in that context, Agilent is happy to collaborate with the people who are interested to make a guide RNAs, right? And we already had some guide RNAs of different disease. Like if you have the cancer panels, we already have these are the genes and these are the guide RNA we make. Already have some, and there are multiple panels like mitochondria, gene expression, protein membrane. These are the panels we already made. We already cataloged back panel. You can use that one, but again how it's going to work and how it's going to screen, it is a little difficult task. So in summary, means you can use the NGS application to identify the causation, what's the cause of that, and I gave the little brief idea how the CRISPR-Cas can be best solution for the drug validation and personalized genomics. So this is This is something you can use the NGS application to identify the problems, and CRISPR might be used for the treatment part, right? So now I'm covering a five slide from the IVF segment. So in reproductive medicine, the most challenging part in the IVF is that aneuploidy in the embryos. Whenever couples go for the IVFs and they do the in vitro fertilization, and 70% are the embryos are basically aneuploidy, right? And so what doctor do, at least in India, what doctor do, they look the good looking embryos, identify it, and basically implant like 3 to 4 embryo, sometimes 2, sometimes 3, depends. So the challenge is that when doctor do this thing, if the good looking embryo is aneuploidy, not going to implant, so your IVF cycle is fail. If it is 2 embryos are good, so both going to implant, it's going to give twins. If it is 3 good embryo, it's going to go 3 kids, right? So it's a challenge. You get either 1, 2, or 3. There's no control on that, right? So we are discussing with the IVF clinic and working on a single embryo transfer paradigm. Means check the embryos, they are good enough, they are euploidy embryo, not aneuploidy, and only one single euploidy embryo is basically go for the further IVF cycle and for the implantation. So we are discussing this thing with single embryo transplant. Doctors basically they do the blastocyst culture, and they don't do, they do the biopsy and frozen of the embryo, but they skip this part, PGS, pre-implantation genetic, in India. And after frozen, these embryo taken and go for the IVF. So they use 3 to 4 embryos and directly implant. What we are talking to doctor, do the PGS, identify the good embryos, once good embryo, and implant process for the implantation. So the couples going to get only 1 kid, right? So when we talk about the problems, if the pregnancy goes with the two twins, there's a lots of problem with the preterm labor, preeclampsia, and it's high rate of the prenatal death in the twins, right? So that's why we talk to doctor, go for this test. Means you can go for frozen, with that do the PGS. PGS means you just go for the screening of all 24 chromosomes, identify all the chromosomes are good enough, identify the euploidy embryo, and then go for the IVF. So how basically they do? By transferring the 1 embryo, basically it also is cost effective for the couples also. It's 5 times less pain basically to grow the kids, right? So how they do? This test is basically done by the biopsy of the embryo. So this is the embryo, and this is the blastocyst, and they take the 1 cell of the embryo, only 1 cell of the embryo. So this is 1 cell they collected. So when this biopsy is done, 1 cell is collected from the IVF embryologist, and for that, because 1 cell has a very low amount of DNA, you cannot do anything with that. So we do the whole genome amplification to increase the quantity of DNA, right? And we label with this DNA with the Cy3 and Cy5 type, right? And hybridize on a microarray slide and go for the analysis, right? If there is some mutations, this is the mutations. If some mutation on the chromosome number 3 or chromosome 22 or deletion at chromosome number 21, it shows that these embryos are aneuploidy and do not process for the IVF, right? So you can identify this by adding the PGS test. You can basically identify the aneuploidy in 8 hours, and and after do this, basically the success rate of the IVF basically increase. When they don't do any test, the success rate going to be 80% with this single embryo transfer paradigm, right? Because he identified all the chromosome, where's the problem. If there is deletion, don't process for the IVF. So this is the smart ART, right? Before to doing process for the aneuploidy, just do the PGS, validate this embryo are good enough, and then go for the IVF cycle, right? Thank you.
All right, so I'm sure after listening to today's application based lecture, you must have found this very interesting, and you saw that how the whole exome sequencing kit can be used for diagnosis of rare diseases, and how the results can be used to choose the right treatment. I'm sure this is just one of the success stories of many things which can be done on various type of NGS based platforms. Dr. Jaiswal also briefly gave you an idea about CRISPR-Cas technology, which is one of the much talked about gene editing technologies available. And I hope you have enjoyed not only today's lecture, but also the series of lectures which we had in the last couple of days and week about NGS technology platforms. And this is one of the revolutionary technology which is really transforming the way we have seen the medicine, and clinics are really getting revolutionized with much faster pace of assays coming to the clinics for the patient care. So your understanding and your knowledge about these applications and these novel technology platforms are definitely going to be very useful. And I must say there is a wealth of data available now from various type of genome sequencing projects. If you know what you are looking for, you can do a lot of data analysis from yourself. I'll give you one instance, one example. The Cancer Genome Atlas TCGA is one of the good resource for looking at the patient's cancer data available. And while they published that work a couple of years ago in Science and Nature, series of papers published. But what is more important, when they made data publicly available for thousands of patients' genome data, then the metadata analysis from that data, many people have looked at very specific type of questions. What is the impact of given genes in patient survival, for example, or looking at specific pathways. And maybe hundreds of papers are actually published just by looking at data alone, not by generating data, right. So what I want to convey you is that you need not to generate the patient derived genome sequence data just for the sake of looking at everything biologically. If you are interested, you can just go download these data, use many of the publicly available software and resources, analyze in your own manner, and then probably you can get some very meaningful and new information even possible just by looking at these data for addressing certain questions. So I hope some of these exposures what we are trying to provide you is going to really make you more comfortable and also make you more enthusiastic and motivated to really take lead forward. Well, I'll thank you to stop today's lecture, but we will have more exciting things to continue in the next lectures as well, when we talk about a new resource for you, which is Human Protein Atlas. Thank you.